All right, let's talk about viruses and the immune system. So imagine you wake up one morning and you're in bed and you're sweating, but you're cold and you're shivering and your neck aches and your head aches and you can't breathe through your nose and you're all plugged up. Well, chances are you have been invaded by a germ of some kind. And what's happening with your fever is that is your immune system's response to this invasion. And by having your body reset its temperature and causing a fever, that releases interleukin 1, which raises your temperature, which then increases the production of the T cells or the lymphocytes, the cells your body uses to destroy the invader. And also, it intensifies the activity of the antiviral protein called interferon, which then helps to fight off the virus as well. But in addition, by raising your temperature, you are increasing your metabolism and your heart rate, and this increases the flow of blood throughout your body, increases the speed at which you're burning energy to give you an increase in energy to fight the invasion. Now, I know when you have the fever, you feel exhausted. Even though you are getting this increase of metabolism, you're burning more energy, but you're also burning up more energy, and you're using it to fight the infection as well, so you feel more exhausted. Now, a germ or a pathogen or an antigen, typically these are bacteria and viruses, and we're going to focus on viruses here. And viruses are interesting because they're not really alive. They have a protein coat, they have DNA, but they don't have the ability to do their own metabolism, and they don't have the ability to reproduce on their own. So what they do is they invade a cell and they hijack all the living functions of a cell to use those to make new viruses and reproduce itself. And there's some different examples of different ways that this happens. One is called a bacteriophage, which is a virus that invades bacteria, actually. So yes, other organisms get sick, even bacteria can get sick. And the bacteriophage kind of looks like a little spaceship, and it attaches to the virus or to the bacteria, and it injects its DNA into the cell like a syringe. And then that cell incorporates that DNA into its own cellular DNA. And when it reproduces itself and makes more copies of itself, and then it starts to transcribe and translate and replicate that DNA. And when it reads the DNA to make the proteins that it would normally make, it now starts making copies of the viruses by mistake instead of the proteins that it would normally make. So the bacteria has been hijacked by the virus, and it continues to reproduce these until eventually the cell is so full of the virus protein coats with the DNA inside of it that it explodes. It's called lysis. And now this virus can go off and invade another cell and start the whole process over again. If you're sick, very possibly you might have influenza, and this is a virus as well. And this is a virus that works a little differently. Instead of injecting its DNA into the cell, it attaches to the side of the cell and gets absorbed into the cell. And then once inside of the cell, then it injects its RNA, it doesn't have DNA, it injects its RNA into the nucleus of the cell. And then the RNA gets replicated by the normal cell processes. And then that replicated messenger RNA gets taken out to the ribosome and translated into the protein. But instead of, again, just like in the bacteriophage, instead of now translating that RNA code into the protein that the cell is supposed to be making, it mistakenly makes new influenza viruses, and then they leave the cell and go and invade another virus. There's another kind of virus, category of virus, called a retrovirus. And HIV, which causes AIDS, is an example of a retrovirus. And in a retrovirus, the virus will attach to the side of a cell and then invade the side of the cell. And the virus uses reverse transcriptase to use a viral RNA to make double-stranded viral DNA. And then the viral DNA gets integrated into the host chromosome. And then the viral DNA is transcribed by the host cell, just like in the other ones. And when it's read and transcription takes place and then translation takes place, that DNA is read, transcribed, and creates new proteins, new viral proteins, which then exit the cell and head off to invade another cell. So in all the cases, they're using maybe different pathways, RNA, DNA, invading, just injecting DNA versus absorbing its whole virus protein coat into the cell. But the end result is always the same. It is hijacking the cell's processes to take over the cell process and make, instead of new cells and whatever proteins that cell should be making, to make new viruses. Now your immune system is responding to this, and it's making antibodies, which the cell makes to attach to the outside of the antigen. And those antibodies are a tool that the cell uses to stop the process. And one way of doing this is to bind to the surface of the virus or the antigen with these antibodies, and they act sort of like a fire blanket. They totally cover it up, and they make it inert. They make it from being able to interact with the atmosphere or the ecosystem or the location, which in this case is the cell. Another thing it can do, instead of just covering it up and kind of isolating it, is it can mark it for the leukocytes, or sometimes called phagocytes, and mark it so that then they can recognize it. And these are big cells that will identify the microbe, and then once they've identified the microbe, they will engulf it and pull it into their cell, and then they will put it into a lysosome, and then they will crush it up and break it up and destroy it, and then they'll spit out the waste, which is this broken up protein, which protein coat, which is no longer able to infect another cell.